Okay, I just got a message that the meeting is being recorded, so I need to think carefully about what I answer. My PhD was completely unrelated to biology or to neuroscience. And then you mentioned that there was a postdoc at the Kavli Institute for Theoretical Physics in Santa Barbara. And they have cookies and tea every day at, I think, 3 p.m. I don't remember for sure by now. And in one of these cookies and the tea meetings, I started to chat with Ila Fit, who was also a postdoc there. And she told me about the discovery of grid cells that was just made at that time. And that completely amazed me. And in some sense, we started to collaborate. I'll mention some of the works that we did together early on at that time. And I just was caught in the field, and in some sense, I never looked back. Is that a good answer?
Certainly.
Okay. So
yeah, we move to your talk.
So let's go to my talk. I need to share my screen, I guess. Let's see if this is working. Okay. All right, so I will tell you about a question that has been occupying me and some of my group members in the past 2 or 3 years. But before I get there, I didn't want to assume that everybody in the audience necessarily has a strong background in theoretical neuroscience and in systems neuroscience. So we'll start with a fairly basic introduction in which I will present some basic concepts. And for those, I know many people in the audience are familiar with spatial representations in the brain and with attractor networks, so please bear with me. After I do that, I will talk about some basic features of dynamics and coding in the entorhinal cortex, and then I'll get to the main topic of the talk, which has to do with coupling between attractor networks in the entorhinal cortex in the hippocampus. And the works that I will present, the new works that I will present, or fairly recent works, were done by Noga Moshaev, who did a PhD in physics with me and is now in Pittsburgh, and by Chagai Agmon, who is still working on his PhD in computational neuroscience in my group. So I always start my presentations related to grid cells with this movie. Even if you saw it, I still find it remarkable every time that I see it. There's a rat here, and the rat is running around in an arena which is 2 by 2 meters in size approximately. There's an electrode implanted in the entorhinal cortex, a tetrode implanted in the entorhinal cortex of this rat, and it's picking up the activity of a single neuron there. And whenever this neuron emits a spike, we draw a little white diamond at the position of the animal. And you're going to see that there's a pattern, that the neuron fires selectively when the animal is in particular locations in space. And in a moment, this will run in fast forward, and you'll start to see the pattern. Quite remarkably, the position of the animal when the neuron spikes are arranged in a very geometric pattern, which tiles the space and has the structure of a triangular or a hexagonal lattice. So this cell is called a grid cell, and there are many cells like this in the rat's brain. They're part of a larger system that contains other types of cells that are involved in the representation of the animal's configuration relative to the environment. And I'll just mention 3 of these types of cells that have been identified over the years. So in the hippocampus, this a seahorse-shaped structure depicted schematically here. Many of the cells have the property that if you record, if you put the animal in an environment and let it run around, you find that the cells fire typically in an environment that is not too large in a single area. There is a single compact firing field. So here you see the firing fields of multiple cells in the hippocampus. And I think about very roughly speaking, maybe about 30% of the cells will have this property in a given environment. And then the medial entorhinal cortex is a part of the entorhinal cortex, which is reciprocally connected to the hippocampus. It both projects to the hippocampus and receives inputs from the hippocampus. So the details in these fairly complicated diagrams are not going to be important for the talk, but the fact that the entorhinal cortex interacts with the hippocampus is important. And the entorhinal cortex also receives massive connections from the neocortex. And in fact, it's the gateway through which the hippocampus interacts with the cortex. And so in the medial entorhinal cortex, these grid cells that I described are found, and these are cells that are, as I mentioned, they fire selectively as a function of the position of the animal, and at least in square rectangular environments, the firing patterns are, the firing fields are arranged on a hexagonal lattice. And I should mention one other type of spatially selective cell, which is found in the subiculum and also in some other areas. And these are head direction cells that modulate their firing rate depending on the orientation of the head relative to the environment. So one particular cell might have a firing rate that depends on the head direction in the way that you see here, and other cells will have similar tuning curves, but with selectivity, with a preferred angle of firing, which is might be different than the one this particular cell. All right. So next I want to talk a little bit about attractor models of neural activity. And one of the most influential models was proposed by John Hopfield in the early 80s. And when he proposed the model, at least initially, he was describing it in terms of very simplified dynamics of neural activity. Neurons were modeled as objects that are in one of two states, an on state, an active state, represented here by 1, and an inactive or off state, which is represented by -1. And the dynamics that John Hopfield described were dynamics in which neurons update their state at randomly chosen times, and when they update their state, they observe the activity of all the other neurons in the network through a linear combination with weights that represent the efficacies of synaptic connectivities. And if this linear combination is beyond the threshold, the neuron goes into the on state. If it's below the threshold, it goes into the off state. And what John Hopfield has shown is that you can select these connectivities such that a certain desired set of patterns form the steady states of the dynamics, meaning that if you put the network in that state, it will stay there. And if you put it in some other random state, it will flow into one of these patterns. So now this model can be generalized to more biophysically realistic models of neural activity, where the internal degrees of freedom of the neuron are continuous and not binary as here. And then you can conceptualize the stored patterns as minima of some energy function. It's wells in an energy function, here shown in 2 dimensions, but this is in the n-dimensional space of neural activity, where n is the number of neurons. And this is not just an analogy. At least for some models of neural activity, you can formalize this statement and show that these patterns of activity are minima of some energy function. Okay, next I want to talk about another important model in theoretical neuroscience, and this is the ring attractor model. And so in the ring attractor model, we imagine that neurons are arranged on a ring. Doesn't mean that physically they're arranged on a ring, but we associate with each neuron an angle, theta, I for neuron I. And the importance of these angles is that the strength of the connection between any 2 neurons depends on the difference in their angles, and this dependence might look like what you see here. By the way, I hope that you see my cursor. Do you see it? Yeah, okay. So this dependency means that neurons that are close to each other are exciting each other, and then neurons that are further away from each other are inhibiting each other. And then under a wide range of parameters and choices of the dynamics, of the model of neural dynamics, something interesting happens, and this is that despite the complete symmetry to rotations in the way that we constructed the model, the steady states of activity in the model look like this. There's a bump of activity of nearby neurons that are co-active. They're exciting each other through the short range connectivity, and they're inhibiting the rest of the neurons. But because of the rotational symmetry in the model, it should be clear to everyone that if this is a steady state of the dynamics, any position of the bump can be a steady state. So the system has a continuum of steady states, and this can be used for storage of short-term memory, because you can imagine that some input to the network, say sensory input, could be activating preferentially a neuron here, and this would cause the bump to appear in this position. And then we can remove the input, and the bump will stay in place because this is a steady state of the dynamics, and therefore you'd be able to read out the position of the input later on from the neural activity. But from the dynamical point of view, what I want to stress is that the activity patterns that are expressed by this network at steady state form a very low dimensional, in fact, one-dimensional manifold, characterized by one number, the position on this ring. And conceptually, we can think about this manifold of steady states as the minimum of an energy function that looks like this Mexican hat. All right, now head direction cells are hypothesized to be part of a network with this structure. But we need to somehow relate the position of the bump in the neural network to the cognitive variable, which is the animal sense of where its head is pointing to in the environment. And so how does this happen? This could happen in two main ways. One, through sensory cues coming from some computation that the brain does on what is the absolute orientation of the head, which then generates some inputs to this network. And the other possible way that this association could be made is that even in the dark, when the animal doesn't have sensory cues, we know that animals can update their internal sense of position based on their rotation. And in fact, it's possible to build into this attractor network the machinery that allows the bump to move at angular velocity, which is proportional to a velocity input that the network receives. And I won't go into the details of how this is done, but there were various models that have proposed how this is done, and especially this the so-called two-ring attractor model described in detail in this paper is a model that I will work with later on in this talk. Okay, so now if the brain really manages to create a precise association between the position of the bump and the orientation of the head, then if we would measure the activity of one particular neuron, we would see that it has a firing rate that depends on head direction in the way of the form that is shown in this plot. Now, everything I told you so far might sound like some model that was dreamt up by a theorist, and in fact it was, but it might be unclear whether it's really related to biological reality. But in fact, we know now of at least one neural system in the brain of flies that follows very closely the principles that I described, both in terms of the dynamics of the network and in terms of the connectivity of the network and in terms of the existence of the velocity integration mechanism that receives velocity inputs and generates a motion of the bump in response to this input. And I won't talk much more about the system, but just mention for those of you who didn't follow all the talks in this series, that the first talk in this seminar series, a beautiful talk by Larry Abbott, provided some additional insights on the system, and especially on how sensory cues can update the position of this bump. All right, so next I want to talk a little bit about attractor models of grid cells. So going back to grid cells have also been hypothesized to form an attractor network. And I won't describe the details of the network I won't describe the network in the models in great detail. Of several versions of these models. And the most important thing to know is that you can think about them as generalizations to two dimensions of the ring attractor model. So for example, in one particular version of the model, we imagine that the neurons are organized on a neural sheet, and the connectivity between them depends on their distance between them, now in two dimensions. Nearby neurons excite each other, neurons that are a little bit further away from each other inhibit each other, but the inhibition is limited in range. And then the outcome of this is that at steady state, the network activity goes into a pattern, becomes a pattern that looks like this, but the pattern can exist in different versions, which are translations, rigid translations of each other. So there's a two-dimensional manifold of possible activity patterns at steady state. And now if the brain generates a precise association between these different versions of the pattern, these different shifts, and the position of the animal in space, then if we would measure the activity of a single neuron, we'd find that it has a selectivity to position that looks like this. But what I want to stress is that while the recordings that the experimental recordings that our experimental colleagues have generated in the past 15 years from grid cells were mostly from single cells and sometimes from pairs of cells, the stronger predictions of the attractor model are not about the response patterns of a single cell as a function of position, but on the joint activity patterns of many cells. And even though recordings were limited up to very recently to at most say, pairs of neurons, the model has withstood some very important tests in the past 15 years or 10 years. And I want to mention just two of these tests. So one of them is that the model predicts modularity, meaning that there should be many neurons that participate in the same attractor network and should therefore have the same spatial response pattern up to shifts. And this has been decisively validated in 2012 in a work from the Moser lab in which dozens of cells were characterized in the same animal. And you can see that in this particular animal, the grid spacing, the distances between the firing fields of grid cells did not form a continuum. There were discrete values of the spacing here, 39 centimeters, 48, 65, and 98. And furthermore, all the cells that share the same spacing, which have been called a module, also share the same orientation of the pattern. Okay, so this is one prediction. Even more importantly than that, the model predicts that if we look at the pairwise activity of neurons, there should be some pairs of neurons that always either fire together or are silent together. And then there are other pairs of cells that should never fire together. And this feature should be very robust to various manipulations of the system. For one thing, moving the animal from one environment to another. And this has been verified, but also other manipulations. So this prediction has withstood numerous tests under manipulations that are pharmacological, genetic, optogenetic. In many of these manipulations, the inputs to the entorhinal cortex are disrupted. In some of them, the spatial response patterns of single neurons are disrupted, but nevertheless, the relationships between the firing of pairs of cells are maintained. Now, when I say that they are maintained in the sense that if you look at some statistics of the correlation between pairs of neurons, these are similar in all the different conditions. And most recently, this has been verified also in sleep. We're not yet at the stage where we can say that the joint activity patterns of multiple cells are very close to a low-dimensional, two-dimensional manifold with a topology of a torus, which is what the grid cell attractor model predict. But presumably, with the introduction of large-scale neural recording techniques into the medial entorhinal cortex, it will be possible to test this in much more detail. All right, now, at this point, I want to say also a few words about grid cell activity viewed as a neural code, as a representation for the position of the animal. And so let's think about a situation where you observe all the spikes of all the cells in one module, and you try to infer from them what is the position of the animal. So you try to infer likelihood for position X given the activity of all these neurons. Now remember that all the neurons in one module share the same periodicity. And so our inference is going to be ambiguous. We're not going to be able to distinguish between this position and this position and this position. So on a first thought, grid cell activity as a representation of position sounds like a bit odd because it's ambiguous. But in fact, as we discussed, there are multiple modules in the brain with different spatial scales. So the real situation that we should think of looks more like this, where you might be able to infer from all the grid cells in module 1 that the animal is in one of the red positions, from the grid cells in module 2 that the animal is in one of the blue positions, from module 3 the violet positions. In this particular case, there's just one position in this environment which is compatible with the activity of all the cells. So therefore, you can ask the question, what is the range of positions that you can represent unambiguously with multiple grid cells, and we and others argued that this range can be very large. And to discuss this a bit more, I'd like to make a simplification and move to a one-dimensional analogy. So let's imagine that we have a rat moving in a one-dimensional world, and we have these meshing gears that rotate as the rat moves to the right. And the angle of these meshing gears is going to be a periodic function of the position. So this is similar to the state of a grid cell module. And we can think about these two angles, theta1 and theta2, which are going to be periodic, as a representation of the position of the animal. So what can we say about this representation? Well, for one thing, if the radii of the two meshing gears are incommensurate, then every possible combination of angles is going to be eventually accessible. And now, how are theta1 and theta2 going to evolve as the animal moves around? So as the animal moves to the right, both theta1 and theta2 are going to increase. And then theta1 is going to complete a full circle before theta2 because the radius of this meshing gear is smaller than this one. And then it's going to wrap and continue to increase, go back to 0, continue to increase. And we can continue like that. At some point, theta2 will also wrap and maybe pop out. The trajectory will pop up somewhere over here. And we can continue like this on and generate a one-dimensional path through this two-dimensional phase space. Now, at some point, we will need to stop. Why? We'll need to stop because presumably there's some range, there's some precision at which we can measure theta1 and theta2. And so we don't want to be at a situation where we would confuse this point with this point. Remember that points along a line represent nearby positions, but this point and this point might represent very distant locations. So if we were going to confuse these 2 points, that's going to be very bad. So we need to stop with our, continue with our line when the line approaches itself at a certain distance. And if you now imagine that instead of 2 modules or 2 meshing gears, we'd have M modules, then you can realize that we can hope for the length of the line that we're going to be able to fit into the m-dimensional cube to be exponential in the number of dimensions. And so using this, based on this reasoning, both analytic and some numerical computations, we've shown a long time ago that indeed the range of the representation, the range at which grid cells can represent unambiguously positions can be very large. It scales exponentially with the number of modules. This idea is interestingly related to an idea that was proposed a long time ago by Claude Shannon, which at some point he was interested in the representation of variables by analog channels, not in the digital form, which occupied him most of the time. So he asked how, he was interested in the question of how would you, what would be an efficient way to represent an analog one-dimensional variable using a certain number of noisy analog channels in an analog manner. And what he pointed out, for example, if we have 2 channels, that it would be very wasteful to represent the one-dimensional variable in the same way in the 2 channels, because that would mean that the 2 channels will jointly always be at a state which is along this diagonal. And we would never sample a large part of the phase space of the states of these possible states of these 2 channels. So a much better encoding scheme would be to map the variable to a kind of a twisted line that more or less fills up the whole phase space, but never approaches itself beyond a certain distance, which is set up by the noise in the channels. So grid cells are doing exactly the same thing. Grid cell modules are doing the exact same thing, where each module corresponds to one of these channels. And based on this reason, both we and others argued that an important feature of the grid cell code of position is that it has a large dynamic range. Dynamic range is the ratio between the range of unambiguously representable positions, the total length of this line, and the readout resolution. And this dynamic range is exponential in the number of modules and therefore also with the number of neurons. Whereas for a unimodal place cell code, if you need to tile the space by neurons, the dynamic range will only scale linearly with the number of neurons. All right, so now this brings me to the main question that I want to discuss in the rest of the talk, which is we discussed so far two perspectives on grid cell activity. One is the coding perspective that I argue that grid cells maintain a highly efficient combinatorial code for position that has a large dynamic range. And then I also discussed before that the dynamic perspective, where we think of grid cell modules as attractor networks that are involved in maintenance of memory and its update based on velocity inputs. But when we discussed dynamics, we did that only at the level of single modules, whereas the advantages of the grid cell code in the high dynamic range arise from the distributed representation across different spatial scales. And this, once you realize that, you realize that there is a problem. And let me explain it. I'll explain it in more than one way. So imagine that we're now back in 2 dimensions, and we have 2 grid cell modules that are telling us that one of the modules is telling us that the animal is in one of these positions, and the other module is telling us that the animal is in one of these positions. And in this environment, the joint activity tells us that the animal is in this position. Now imagine that each one of these modules is an attractor network, and the animal is deprived for a while of sensory cues, and the attractor network is storing this representation in memory, but the attractor network is noisy, and the noise causes some drifts in the representation. Now it could happen, for example, that the representation in module 1 drifts to the left, and the representation in module 2 drifts upward. And you can see that what happens is that even though these drifts are small, suddenly the joint activity of the 2 modules is no longer compatible with any position in the vicinity of the original position, and it jumps to a faraway location. So if these kind of incompatible drifts can occur in the different modules independently of each other, they could lead to what we call catastrophic readout errors, or catastrophic errors in the position represented in the system. I am going to skip these slides. All right, so in the one-dimensional analogy, remember that we set up our system such that this line never comes too close to each other, so that if we try to read out activity, we're not going to mix or confuse this position with this position along the line. But now the system is also engaged in the storage of memory, and the memory starts to degrade in each one of the angles, θ1 and θ2, then if they degrade in a compatible manner along this line, that's not a big deal. That would cause local errors in a reconstruction. But if the meshing gears are allowed to rotate independently of each other, and they can, and the state of the system can move in this way, that could be catastrophic. So the question that I'm going to address in the rest of the talk is whether there is a mechanism that could suppress such incompatible drifts in different modules. And another way to think about it is that if we have m one-dimensional modules, then we want the phase space of the m modules is an m-dimensional space, and we want the system to be able to sample this whole space in order to have a large representational capacity. But in terms of update, in terms of the dynamics, we want the system to respect the continuity of motion, which corresponds to motion just in a one-dimensional subspace. And we want to avoid motion of the 2 modules, which is outside of this small dimensional subspace. So if we want to move to 2 dimensions, then m grid cell modules in 2 dimensions have a phase space which is 2m dimensional, but we want to restrict local changes, dynamic changes in the states of the modules to sample a much smaller 2-dimensional subspace in order to avoid catastrophic readout errors. So I'm going to tell you about 2 different works from my group in which we proposed 2 different solutions to this problem. The first one involves, which I will talk about first, involves only the entorhinal cortex and connectivity between grid cell modules. And the second one involves also the hippocampus. So remember that our goal is to couple the grid cell modules such that their motion, their dynamic changes will always be compatible, but allowing them to sample all possible states. And the way we approach this, first I'm going to describe everything in a one-dimensional analog of grid cells, but everything that I'm going to tell you maps easily also to 2-dimensional grid cells. So in 1 dimension, we can think about grid cell modules as attractors that are structured as a ring attractor, and the position of the bump on the ring attractor is periodically associated with positions in space. And then we model each grid cell module as a ring attractor, actually a double ring attractor, which is the model that has built into it also the ability to integrate velocity inputs to move the bump in response to a velocity signal. And what we want is that if module 1 happens to move, this will cause a compatible motion in module 2 and vice versa. And so one crucial element in the model was to find a simple way to read out the velocity from the activity of module 1, and I will not describe this in detail right now. Once we have that, once we are able to read out the velocity from module 1, we can inject it as a velocity to input to module 2. We already have built in the double ring attractor model the ability to take a velocity input and use it, and based on that input, change the position of the bump. So we're going to read out motion from module 1 and inject it as a motion generating signal into module 2, and we're going to do the opposite as well. So here you see we have some connection from module 1 to module 2, from connection from module 2 to module 1. And turns out that if that's the only thing that we do, then we create a positive feedback loop. If module 1 happens to start to move, that causes a motion in module 2, which causes back a motion in module 1, and the system just becomes unstable. So in order to tame this instability, we need to introduce also a similar coupling between each module to itself, which is inhibitory. It's a negative coupling. And then if we have m grid cell modules, we have m square coupling parameters. And our goal is to set these coupling parameters such that if one of the module moves, all the other ones will move at an angular velocity which respects the ratios in the grid space, desired grid spacings of the different modules. More precisely, we imagine that each module has a velocity input coming into it, and we want to suppress incompatible motion of the modules of two types. One is incompatible motion that might arise from the fact that the velocity inputs to the different modules are not precisely the same. They might have some noise in them. And the other is that we might want to suppress incompatible motion in the different modules that arises intrinsically from noise within each one of these attractor networks. It turns out that the easier problem of these two to analyze analytically is the first one. So I'm going to tell you first about this goal. And I'm not going to go into many of the details. It turns out there's a family of solutions that solve the problem. And we can further restrict them if we wish, for example, by introducing additional constraints, like, for example, we might require that only adjacent modules couple to each other. And then the solutions that work well involve strong positive coupling between modules and also strong self-inhibition to avoid instability. And I just want to show you that this works. So here we go back to 2 dimensions. We have here 3 grid cell modules in 3 dimensions. We take a trajectory of an animal. We generate from it velocity inputs, which we inject to 3 grid cell modules, but we inject them with noise. So each one of the modules receives independent noise, which is independent of the other ones. And you can see here that if the modules are uncoupled, then each one of them follows a trajectory that has more or less the same shape as the trajectory, the original trajectory of the animal, but there are also some differences. If you look at the blue and yellow curves here, you see that they're not exactly the same. And on the other hand, if we couple the system in the way that I described using the appropriate coupling parameters, the motion in the 3 modules is much more coordinated, as you can see here. Now you can wonder whether the differences that I show you here in the uncoupled case matter. So what we can do is take the dynamics, the simulated dynamics of these 3 grid cell modules, generate spikes from each one of the modules, and then take all the spikes and decode them to try to figure out where the animal is. So in the coupled case, when we do that, we get a decoded trajectory that follows the true trajectory. It gradually and gracefully accrues errors relative to the true trajectory because of the noise in the velocity inputs, but it's continuous and it looks good. In the uncoupled case, we very quickly get these sharp and large deviations of the decoded position from the true position. Sometimes it comes back and it jumps again. And these are exactly the kind of catastrophic readout errors that I described earlier conceptually. And we can quantify this. We can show that indeed, in this case, in the uncoupled case, we have these catastrophic readout errors, and once they start to occur, the mean square error of the decoder increases dramatically. This is a semi-logarithmic plot. Now, the last thing I want to tell you about this model is that it turns out that the same connectivity that confers the system with a resistance to incompatible noise in the velocity inputs also confers it with resilience to intrinsic noise in the different modules. And a way to quantify that is to look at the diffusion tensor that characterizes the random motion of the different modules. And in the uncoupled case, the modules drift independently of each other, so the diffusion is completely isotropic in the m-dimensional space, where it's just two-dimensional. But once we couple the module, the diffusion is really restricted to a narrow direction, which corresponds to the direction which is compatible with local motion in one dimension or in two dimensions when we're dealing with two-dimensional grid cells. So to summarize what I've told you up to now, we discussed the fact that the grid cell code is sensitive to incompatible drift in different modules, and this is a problem if we want the grid cell system to represent position, but also store it and maintain it in memory and update it based on velocity inputs. And it turns out that a simple form of a coupling, synaptic coupling between grid cells allows for stable memory maintenance because it suppresses incompatible motions. So on one hand, we have a large representational capacity, that we have these M grid cell modules that have a large phase space that is all of it is accessible in principle, but local motion is restricted. So we can think about the system in general as a way for the brain to represent a low dimensional variable with a very high dynamic range, while not just representing it, but also allowing for a distributed representation, which is also resilient, which is also compatible with maintenance of memory, and in a way which is resilient to noise. Now, in terms of predictions, I would say the most broad prediction would be that this model suggests that there could be a reason for grid cells in the entorhinal cortex that belong to different modules to be synaptically connected to each other. And at this point, we don't know whether that's the case or not. Okay, so next I'd like to talk about a more recent model, work with Hagai Agmon, which is concerned with the hippocampus. So the idea, one of the ideas in this work was that the coupling that I described earlier could also be implemented through a connection with the hippocampus. So the idea is that the hippocampus could read out the position from the joint activity of multiple grid cell modules. And even though we argue that the representation of position in the hippocampus is more coarse, it's less precise than in the medial and entorhinal cortex, it could be sufficient to feed back to the medial entorhinal cortex and fix incompatible drifts in the different modules and avoid these incompatible drifts. Now, this idea is not a new idea. It was explored previously, but not at the level of a detailed network model of attractor dynamics. And furthermore, there's another issue that has been largely overlooked, and that has to do with the way the hippocampus represents positions across multiple environments. So, which is more complicated than what grid cells are doing and what head directions are doing. If you record from several places in the hippocampus in one environment, for example, you might see that the red, blue, and green cells fire in these locations in environment 1, and then you move the animal to another environment, you find that the spatial relationships between the positions in which the cells fire are not maintained. So these two cells, the green and the blue, which fire at nearby locations in environment 1, will typically fire at faraway locations in environment 2. And this is called global remapping. Now, if you think about this in terms of the population activity patterns of many cells, this means that the hippocampal code or the hippocampal representation doesn't only represent position in a given environment, it also represents in which environment the animal is present. So for example, if you would observe the joint dynamics of the cells, we might be able to infer that the animal is in this environment and not in this one because the green and blue cells are firing together. They cannot do this in this environment. Now, There is evidence for attractor dynamics in the hippocampus, but this tells you that the attractor dynamics has to be more complicated than what we discussed in the context of the ring attractor model or the models of grid cells. And if ring attractors have a manifold of steady states that lies on a ring, and grid cell models have a manifold which is like a torus, if conceptually, if placers form an attractor network, this network on one hand has a continuum of steady states lying on a two-dimensional manifold for each environment, but it has distinct manifolds for different environments. So this is in some sense like a combination of the Hopfield model where you have discrete wells, but each one of these wells is a continuous two-dimensional manifold. And I'll talk a little bit about how this is implemented in a neural network in a moment. So one of the questions that Chagai and I wanted to answer is whether it's possible at all to couple all these attractor networks, the multiple grid cell networks and the hippocampal networks, such that they will represent coherently a position in two-dimensional space. And another motivation for the work is that if you look at theoretical works on grid cells and placers, you see that there are quite a lot of works that ask how placers might be generated from grid cells, and there are quite a lot of works that proposed ways in which grid cells could be generated from placer inputs. And indeed, these two systems are reciprocally connected to each other. But as I emphasize in this talk, we think that there's also a very important role for recurrent connectivity in each of these networks. And we think about them as attractor networks. And so we think that it doesn't really make sense to ask whether grid cells drive placers or placers drive grid cells. Our point of view is that these are probably attractor networks that are reciprocally connected to each other bidirectionally. And the questions that we wanted to answer is whether, first of all, it is possible to set up a large attractor network composed of these different sub-attractor networks, such that they will always represent position in a compatible manner, but would do that over multiple spatial maps compatible with what we know about remapping of places and grid cells in different environments. And then the question is whether, once we set up such a network, there will be some interesting emergent consequences. So here's the architecture that we worked with. We imagined that we have distinct attractor networks for each grid cell module with the connectivity that supports grid cell dynamics in each of these networks. We have one network of places which allows for the representation of multiple spatial maps, and I'll talk about that in a moment. And then we also generated some bidirectional connectivities between these two systems. So before I talk about this bidirectional connectivity, I just want to point out how one can set up multiple spatial maps in an attractor model of places. And so let's first talk about how one generates an attractor networks of places in one environment. That's easy. You can imagine that you set up synaptic connectivity between places that depend on the distance between the centers of their receptive, the spatial difference between the centers of their preferred firing locations in space. And this connectivity could have local excitation between nearby cells and very broad inhibition. And if you set up a network like this, then you can easily get a network that expresses a steady state bump patterns in the space of the cells when you order them based on their preferred firing location. Now, if we think about some environment, other environment mu, then to set up the connectivity that would support the joint dynamics of cells in that environment, we just need to take the same kind of connectivity, but set it up as a function of the difference between the firing locations in that particular environment. Now, if we imagine that the relationship between spatial firing locations in different environments are just random, then we can think about this W mu, the connectivity matrix in W mu, as obtained from this connectivity matrix just by applying a random permutation to the rows and columns of this matrix. So now we have a matrix, a synaptic connectivity matrix that represents, that could generate an attractor dynamics in each one of the environments, and then we just simply add them up. Very much in the spirit of the Hopfield model. There is some correction term that I won't get into here, but it turns out that when you do that, and if you don't try to embed too many maps in the environment, you end up with a system that has steady states that look like localized bumps of activity in each position in each one of the maps, meaning that if you would order the cells based on their preferred firing locations in one of the maps, then you'd see a bump of activity, but it could be anywhere and it could be in any one of the maps. And this idea is an old idea proposed by in these works, and more recently, Sophia Rossay and Remy Monasson has been further exploring this idea. And so we took this idea and used this architecture and used it to model place cells. But then we also generated connectivity between these place cells and grid cell modules. And I won't say much about how we set this up. I'll just say that the strength of the connection between a place cell and a grid cell was set to be proportional or linear in the overlap between the desired spatial receptive fields of the cells across all possible positions and across all possible environments. And I just want to show you first that this can work. So we do this now in a one-dimensional analog of the system. And I'm showing you here the joint activity at a given time of all the place cells and all the grid cells in our system. So the place cells are shown in black. And we're ordering all the cells here based on their preferred firing location in environment 1. And so you see that the place cells form a nice bump of activity localized around a certain position. For the grid cells, I should clarify that the grid cell that fires here is actually the same as the grid cell that fires here and here and here and here, because grid cell firing is periodic. So you see each grid cell 5 times here in this module, or maybe 6 times and 5 times for the red module, and so on. But you see that you have a localized bump of activity also in the 3 grid cell. You have here 3 modules. So you have a localized bump of activity in the 3 grid cell modules, and all these bumps of activity are coordinated with each other. And this is a steady state of the dynamics. And we have other steady states that correspond to other positions of the bump. Now, if we would look at this joint activity pattern from the point of view of environment 6, just reorder the cells based on their preferred firing locations in environment 6, in the grid cell module, we will just see a shift. But in the place cell network, because the relationships between preferred firing locations are random permutations, we'll see just a scattered activity pattern. So we have all these steady states which correspond to different positions in environment 1, but crucially, the network also has steady states that look like this, where when we order the cells based on their preferred firing location in environment 6, we see localized bumps of activity, which are coordinated in all the networks. And then we can do many tests to see that we have a representation of all positions in all environments, and we don't have some spurious states that don't adhere to these rules. Okay, so now I will very briefly talk about some of the consequences coming out of this model, and I'm soon running out of time, so I will do this quickly. One consequence is that you can implement velocity integration in the grid cell network and not in the place cell network.
You are fine.
Yeah?
You're fine. Yeah.
All right. So we can implement velocity integration in the grid cell network, and it's easier to do it in the grid cell network because the spatial relationships between grid cells are maintained across different environments, which is not true for place cells. So you can implement velocity integration in the grid cell network, and because of the connections between grid cells and place cells, the activity in the place cell network follows the activity while the grid cell modules are integrating velocity, the activity in the place cell network follows what the grid cells are doing. Second is the goal that I described as the main goal of this talk, which is to couple the grid cell modules. Because we have the connections between grid cells to place cells, and we also have a back connection from place cells to grid cells, it turns out that the modules are coupled. If we don't couple the modules, and we just simulate the dynamics of the grid cell modules as noisy networks, the state of each grid cell module performs a random walk, and gradually they move around in an incompatible manner with each other. And once we introduce the coupling to place cells, the state of the grid cell module still drifts around due to noise, but it's in a compatible manner. This is much, much less detrimental from the point of view of representation and decoding. And this works across all the maps that are embedded in the network. Next, I'll describe something that we just found to our delight that happens in the network, which is that the network, the model recapitulates a surprising observation that was made 3 years ago in the lab of Cliff Kentros at NTNU. What was done in the experiment is that Kentros and his colleagues looked at what happens in the hippocampus when the entorhinal cortex is manipulated. And the manipulation was done by DREADS, designer drugs, designer receptors activated by designer drugs, which were introduced, expressed in the medial entorhinal cortex, specifically, not specifically in grid cells, but in layer 2 stellate cells in the medial entorhinal cortex and very locally in the medial entorhinal cortex. What these DREADS were doing once activated was that they either hyperpolarized, meaning suppressed grid cell activity, or depolarized grid cell activity. And it turned out that when grid cells were hyperpolarized, nothing very interesting happened in the hippocampus. But when grid cells were depolarized, meaning that they became more active, suddenly something very interesting and surprising happened. Some of the places maybe changed their firing rate, but kept firing before the manipulation and after it in the same location as they fired before. But other places abruptly shifted their location of firing from the original location to a new one. And it turns out that our model has exactly the same phenomenology. And we relate this to the fact that, so what we see that happens in our model is that if we apply depolarization to the grid cells and look at the place cells, if we apply a strong enough depolarization, you can see here that after the depolarization, we still have a bump of place cell activity at the original position, but we also have some place cells that are suddenly firing outside this bump. And if we look at the same activity pattern from the point of view of another environment, we actually see that there is a bump of activity also in that other environment. So what this tells us is that the activity with strong enough depolarization in the grid cell network, the activity patterns that are expressed in the place cell network are mixtures of activity patterns that previously represented unique environments. I think that I'm going to skip this slide, which just quantifies that more precisely, and just mention that we then relate this phenomena, the expression of these mixed patterns, which is expression of mixed patterns is also occurs in other attractor models like the Hopfield model. We relate the emergence of these mixed patterns to what happens to single cell activity as a function of position. And we find that we see the same phenomenology that was observed by Cliff Kentros. So we put the system in a state that represents one position in one particular environment. Then we apply the depolarization of the grid cells, and then we start to path integrate in order to sample the whole space. And we find that for some cells like this one, the one that you see here labeled G, or the one labeled age, the cell keeps firing at the original position, but other places shift the position to a new location. All right, and I think I'm really running out of time, so I will just mention briefly another emergent consequence of the model, which is related to an observation that has been made by several groups in the past 3 years or so, which is that grid cell firing fields aren't precisely periodic. If you try to measure them, and you, for example, in this case, you see that this grid cell, this is now the firing of a single cell as a function of position. You see that it's firing more strongly here than here. And this is not just statistical, coming from statistical errors of measurements. It's consistent to take the animal out of the environment and put it back in. And our model also predicts that phenomenon, at least to some extent. I think I won't go into the details, but just say that it's related to the fact that at different firing locations of the same grid cell, the place cell network is at a different state. and the inputs coming from the place cells to the grid cells associated with other environments are different in these different locations, and that causes a variability in the firing rate. So I'll just sum up what I've shown you here, which is that we proposed a way to connect attractor networks that represent grid cells and place cells such that they support a joint coordinated representation of position over multiple environments. And then we showed that there are several consequences. I mentioned two functional consequences: the ability to perform path integration in the grid cell network, and the fact that it carries over to the place cell network, the fact that grid cell modules are dynamically coupled to each other. And then I mentioned some emergent consequences, which may explain recent experimental observations. So I'd like to now end and acknowledge people whose work I mentioned today. So some of the earlier works from a long time ago that I mentioned were works that I did in collaboration with Ila Fit, mostly when both of us were postdocs at Santa Barbara. And the more recent works that I mentioned, again, were done by Chagay Agbon and Noga Moshaev. These days, we collaborate with several experimental groups on various questions related to grid cells and also other topics. These are groups that work on, with whom we're working on questions related to grid cells. And I just want at the end to advertise, to put on an advertisement. We're very excited to start a 6-year collaboration with the Moser Lab, funded by the ERC Synergy program. And we're looking for, we're hiring postdocs and graduate students. So, and we're looking for, obviously, for people with very strong analytical and computational skills, but we're also particularly interested in people who are interested in relating theoretical work to experimental research in systems neuroscience. So if anyone here is interested, or if you know people who might be interested, you're very welcome to look at this link here. And with that, I will end.